Otherwise, we're going to move into chapter 3. Now, before we move into chapter 3, I just want to let you know that your first exam is next Monday. Yeah, I know. It's fast and furious because we have this weird Labor Day break. Next Monday is the 4th week. Okay? What you will have to know for the test next Monday is your lecture notes. Did you start the video? You're so good. Thank you. The lecture notes. Just the lecture notes will be on the lecture exam. Chapter 1, 2, 3, and a little bit of part 4 called epithelium. I think I wrote it up on the board there. Okay? So chapter 1, 2, 3, so it's not going to be that bad. It's only 3 chapters and a little bit more. Okay? And epithelium. So we do part of the tissue chapter. Right? Okay? So once again, you should be studying an hour or 2 a day, getting through the material, trying to memorize the notes as best as possible. Okay? Yeah? Okay. So, chapter 3 we're moving into, we're going to look at the cells. Chapter 1 was that nice, easy introduction. Chapter 2 was chemistry, which once again is a little bit disjointed, but we're trying to teach you bits and pieces of what you'll need to know rather than everything about chemistry, right, which you could learn over years, really. So now we're going to look at the cells themselves and how they interact with each other. And believe it or not, this is actually a typo. It should say trillion there. I would never test you on that, but just as a side note to let you know how many cells we have in the body. Amazing. All right, so why do we have to study cell? Right, let's just get into the fun stuff. Well, I think this is the fun stuff, but anyway, I'm a dork too. That's a whole different story. So we study cells because we're made up of cells, right? And it's the functioning of those cells which create the functioning of us. So we do need to know the basics of these cells and then how they're put together into tissues, okay? It obviously is the smallest form of life. I call them little mini-me's because basically they're structured similar to us. And there's little organelles, which are tiny little organs that provide very similar functions to us as a whole. So if that helps you to assimilate or associate a cell with us, maybe it'll help you remember some of these things. Now, the basic structures of this cell. We have that plasma membrane, which we're going to look at the specifics of what make up that plasma membrane a little bit, because that is a huge, believe it or not, that plasma membrane is the hugest factor of how it interacts with our environment and also how cancer cells hide, is that plasma membrane. Because think about it, right? Cancer cells are cells that are not normal. Why doesn't our body find them? Right? What does our immune system do all day? Looks for abnormal cells. Well, what happens? Well, those cancer cells hide behind that plasma membrane. I'm going to show you how that happens. Okay, so plasma membrane. We have eukaryotes versus prokaryotes. You are eukaryotic, has the plasma membrane, the nucleus, which is the brain of the cell, and cytoplasm, which is the gunk in which those organelles or those mini organs sit. Okay, we're not going to talk about prokaryotes at all. If you're taking bio, you go over that. We're not. We're going to move right on. Okay? Now, something that is important for you to understand, you guys are already seeing it if you're in clinical, is fluids of the body is so crucial. What happens if you go into the hospital and you're really dehydrated? They put in an IV. They have to reestablish the right amount of solutes in the solvent or solution that's in your body for your body to function properly. So we need to understand where those fluids are in and outside of cells. So intracellular will obviously be the fluid inside, and it has a very specific chemical balance. I'll give you some of the basics of that. Extracellular will be the fluid outside of the cell. Okay? We might use intercellular. So I said intracellular, I said extracellular. Intercellular just means the fluid between them. I don't use that much. I want you to know intracellular versus extracellular fluid. Okay? Because we need to talk about how that moves in and out of cells. Right? Does that make sense? So the solution around a cell needs to pretty much balance the solution in a cell, right? Otherwise, what's going to happen? Yeah, either fluid can move into a cell or out of a cell. Does that make sense? So we need to have a nice balance of those fluids in and out. Does that fluid always stay the same in and out? No, of course. It changes a little bit. Well, nutrients come in and wastes go out, so it is changing. But overall, we're going to keep about the basic solution content and concentrations of solutes, right? So cells are like little people. They remain small. When we remain small, what we're doing is we're increasing surface area. When we increase surface area, we have more interaction with the environment. Now, cells have surface specializations. There's microvilli, okay? Now, microvilli are not cilia. Do you know what cilia are? A lot of people know what cilia are.
So the hair.
Yeah, they're hair-like projections that stick out of the top of cells. And those cilia live in our respiratory tract, for example, other areas of the body. And what do they do with junk from our lungs?
Smell it, get rid of it.
Right? They move it right along and they get it out of the lungs because we don't want junk in our lungs or we could get pneumonia, right? Now that's different from microvilli. Okay, microvilli are thicker and they provide more of a wavy-like presentation. Okay, anytime I do this, I'm going to do it a couple times. This wave-like presentation, like having small cells, this wave-like presentation increases surface area. So we'll see, for example, the cells of the GI tract have microvilli. Because in the GI tract is where we absorb all those yummy foods that we just ate for breakfast. And we want lots of surface area to absorb as much as we can from that foodstuffs. Does that make sense? Okay, so cilia, microvilli. The last thing we're going to talk about is glycocalyx. Okay, glycocalyx is this slimy, sugary layer on the outside of cells, which helps cell-to-cell recognition. And one of the theories with cancer is this glycocalyx on cancer cells can actually hide them. And that's how they can survive. Okay. We also use these surface specializations for bonding of cells together. I'm not going to get too much into that, but those I want you to know. And cilia, I know, are a little further in the notes, excuse me, but you can pop cilia down there just so you remember hair-like structures. Okay. Any questions so far? Okay, so different cells. I don't like giving you laundry lists. This is a laundry list, I know. Point and shoot. We're going to learn all these as we go. And I'd rather teach it to you bit by bit so you can see where they are and what they do. But here's a picture to give you a little introduction to all types of cells. Okay? So let's talk about the plasma or the cell membrane. It is a phospholipid bilayer. And I think I've got that maybe. Oh, maybe not. Okay. Okay. So it's a phospholipid bilayer. Okay. So I have a lipid bilayer there, but actually phospholipid bilayer. And what that means is that you actually have phosphorus and lipids. And those, I know I'm not supposed to draw, well maybe you can see it here. So this is the phosphorus. It looks like a double stick lollipop. I think I talked about it in the second lecture. Did I? Yes. Okay, great. So you have those double stick lollipops, right? Phosphorus is the lollipop and the lipids are the sticks that stick in. And then you have one on the other side, phosphorus and the lipids here. Why do we establish that plasma membrane with that phospholipid bilayer? It makes it
Hydrophobic, right?
You're on the right track. Well, there's hydrophobic and hydrophilic. Hydrophilic is the phosphorus, hydrophobic is the lipid. It makes it semi-permeable. It makes it really selective. It's just like dating, right? You can decide who's going to come visit you, right? Okay, and the way you do that is through these protein channels. Okay, can some things sneak through the phospholipid bilayer? Absolutely. They have to be small or lipid soluble. Okay, are you with me? We have cholesterol that actually is in there to strengthen it, but mostly we're going to talk about these protein channels. These protein channels are essential to letting things in and out of cells. I call them little purple people eaters because in your book they're painted purple. Okay, they're not purple in people, just so you know. But they are proteins that span this phospholipid bilayer. Is everybody with me? Please, the minute you get lost, stop me. Okay, so we can talk about those. Okay, so this is the bridge from outside to inside, determining what's going to pass through. Let's talk about these protein channels. Okay, so we're going to talk about, there are some partial proteins you'll see in the notes that only partially go through. Most of them are integral, which means they pass all the way through that phospholipid bilayer. Okay? And we're just going to focus on those because those are the ones that we need to understand in order to understand how things get in and out of cells. Are you with me? The other thing to understand about these protein channels, guys, is there's hundreds of thousands of them in a cell membrane. And a lot of them are very specific. There are some that are only for sodium. There are some that are only for potassium. Are you with me? There are some that are only for glucose. So it's not like every one of these protein channels will let things in and out. They're very specific. Are you with me?
So those are defined with the substrates, certain substrates?
Yes, and we're going to get into that next. Well, you could bind with a substrate for a second messenger. Is that what you're talking about?
For what?
For a second messenger?
Yes.
Yes, that's what we're going to get into that. Okay, so it's going to transport, it's going to send signals. It can also strengthen the cell or the cytoskeleton of that cell membrane itself. Okay, so transport, simple. Things pass through it, just like cars on a bridge. We need more sodium, you can bring sodium in this way. We need to get rid of potassium, you can send it this way, okay? So you can transport things across, and it's just as simple as that. Now, some of these here you can see can just passively pass through. Some need ATP. So if they need ATP, they are using what? Energy. Remember, ATP are little packets of energy that we have throughout the body that will phosphorylate structures. This was from chapter 2. If I'm saying things that don't ring a bell, make a note of it. Go back and study the videos again. All right? And it primes this protein channel to work. It'll allow it to open and it'll allow things to pass through. We'll talk about one specific, actually a couple specific ones that use ATP in a little bit. Okay? Some of these protein channels simply receive signals. Quite often we'll see this in the endocrine system, the hormone system. These signals will attach to this protein receptor and send a signal into what's called a second messenger. That is a very complex series of steps. That's as far as we're going to go with it. Okay, you just need to know that it'll send a signal into a second messenger. Because don't forget, it's very selective on the membrane. It only lets certain things in. We can actually send signals without even sending things in. That will happen through those protein channels as well. Some of them just help to strengthen the cell and the structure of the cell, which makes sense because it just invests through the width of the cell membrane itself. Okay, excuse me. So just to review very quickly, this is part of that cell membrane with a phospholipid bilayer. I know you can't see that in your notes, but once again, don't forget you can bring the PowerPoints up on your laptop if you want to actually look at the pictures themselves. We've got phosphorus head, lipids, phosphorus head, intracellular fluid. The cell is constantly pushing things in and out because it's just like a little person. It needs food, it needs oxygen, it needs to get rid of waste, right? We have a selectively semi-permeable membrane. Many of the things will pass through these protein channels, either through channels themselves or through second messengers, okay? Is it all making sense? Okay. Right. So, we need to have some connection between these cells, right? Otherwise, they would just float out in space or fall off our bodies. So we have 3 types of membrane junctions. Tight, which is tight. It's in areas that we don't want to let things in. I'm going to give you an example in just a minute. We have desmosomes, which are a little looser. Are they still somewhat tight where they are? Yeah, but they're a little looser and they're more like spot welded in different areas on the edges of the cells. And then gap junctions. These actually form a tunnel from cell to cell. Okay, and I'll give you examples of these in the notes. So tight junctions we're going to find in the gastrointestinal tract. Keep in mind when you go and you eat, you ingest tons of yucky stuff. Lots of yummy stuff too. But along your GI tract, every cell has tight junctions between it. Then the cell decides what passes through the cell to ingest into your system. And the tight junctions allow that level of protection between each cell. All right, so gastrointestinal forms a very tight barrier between cells. I don't know if these pictures help or not, but you can see that the connection runs from the top to the bottom of the cell. It's pretty solid. Desmosomes are those spot ones that are tight where they are, but they're not as often as the other one. Okay? We actually have these in the skin. So it helps to hold the cells together, but we can let sweat out. We can absorb some substances through the skin. Okay, so it's not as tight as the other ones. We find this in, and there's other areas, but these are the examples I'm just going to give you. Okay? And there, once again, you could see just an occasional desmosome as opposed to those tight junctions. The last one is a gap junction, and this actually forms a connection between cells with a hole in it. And what that does is it allows us to transport ions from cell to cell. We see this in the heart. The heart beats how many times a minute? 60 to 100 beats per minute. It's able to do that because of electrical signals and also the transmission of ions from cell to cell in the heart. We transmit these ions through the gap junctions. Okay? All right. There's a little, I haven't looked that up for a while, to be honest with you. Gap junction blockers for migraine, it blocks the transmission of those substances. So, if you want to look at that, you can. And you can see there's that channel from cell to cell. Okay? Any questions on any of that? Okay. So, moving on to, we'll move through these and then we'll take a quick break. How's it sound? So molecules crossing that plasma membrane. We have passive transport and active transport. Active transport will use what?
ATP.
ATP, right? So it's going to use energy. Passive uses no energy. I'm going to give you examples of each. I want you to make note of something. Endo and exocytosis are active transport. So it makes it look like a third thing. I should have moved it over a little bit. But endo and exocytosis are examples of active transport. They use energy. And I have another example as well. Okay. Why do we need to be able to transport substances?
To get nutrition or to get rid of
Yeah.
the dissolved.
So we're looking at the cell. We need to understand how we get nutrients and oxygen into the cell. We need to understand how we get waste out of the cell. But then we're also going to start looking at the movement of ions across cell membranes, which actually help to make like muscles and nerves work. Is everybody with me? So these transport systems will apply to how substances get into and out of cells, is what we're looking at right now. Okay? Are you with me? Okay. So, easy peasy. You probably know this, right? With passive diffusion. Do we know diffusion? Diffusion is how particles distribute throughout a solution evenly. Are you with me? I always think of it as a party. If you go into a party in a room this size and you have this many people, is everyone going to be huddled in one corner? That would be weird, wouldn't it? Right? Is everybody going to be huddled in one corner? No, we diffuse, right? People will move out and separate out and kind of fill the room. Okay, same thing with molecules. If you put a certain amount of molecules in a solution, they'll distribute evenly throughout. Okay, now there's some exceptions, of course, but you get my idea. Okay. Now, osmosis is going to be the movement of water across a membrane. People get confused about this one. So if you have a membrane through which so let's use salt water, just to keep it simple. If you have a membrane separating two concentrations of salt water, are you with me? Okay? And you can move the water across, but not the salt. Water will actually move to the area of what? Higher or lower concentration? Does anybody know? Higher. And I have good pictures of this. I just want you to think first. Higher. And it'll actually balance it. Okay, so osmosis is the movement of water. Because across cell membranes, we could have solutes pass, we could have water pass, right? So both of those things can move to balance the solution. That's the end goal, is to balance the solution. So let's take a look at diffusion through protein channels, facilitated transport. I was looking at this earlier. I had moves 3 ways, but then I only have 2 things with a subdivider. Don't worry about it. We're going to focus on diffusion. We understand protein channels, and those protein channels will help to pass substances through them. Okay, so let's look at some pictures, because I think pictures are worth 1,000 words. This is going to be the process of what? Diffusion. Okay, diffusion. So you see those particles simply diffuse throughout that solution evenly. What's this an example of? Don't look at the answer, try to look at the picture and think about it. A is how it begins, B is how it ends. This is the process of, we just spoke about it, it's the movement of water across that membrane. This is osmosis. Did you ever do this experiment in chemistry where you have the original YouTube? Get it? Anybody? No? Yeah? Okay. So, right, you have that YouTube and you have that membrane through which water can pass. Water will move to the area of higher concentration to do what? Once again, we're trying to
Dissolve or balance.
balance solutions. Are you with me? Okay. Is this making sense? Active transport is going to turn ATP, adenosine triphosphate, into adenosine diphosphate, because you're losing one phosphate to phosphorylate, whatever it is you're giving the energy to. Are there more steps to that process? Yeah, there's a lot, but that's as far as we're going to go. Okay? Exocytosis and endocytosis are active. Exocytosis, I should say, spits stuff out. Endocytosis sucks things into cells. And basically what will happen is your cell membrane of the cell will envelop, I actually have a picture of it, and bring things in, or push things out. It's as easy as that. Now, we're also going to talk about the sodium-potassium pump. And I have a picture that will explain, well, what you should know for now, if you're looking at a resting cell, okay, a cell that's not actively doing anything right now, that cell is going to have more sodium outside. And I'll tell you why in a little bit. Just take my word on it. And how do we write the sodium ion? Na+, right? A resting cell also has more potassium inside. How do we write potassium? K+. Are you with me? Yes? Okay. Why? That's how these cells regulate themselves. And there's reasons why we'll get into. This is a wax on, wax off kind of moment. You ever watch Karate Kid? Right? Just trust me. Learn these basics, they're going to build. Okay? Right? So this is important because we're going to talk about a sodium-potassium pump, which uses energy as well, which helps maintain sodium primarily where? In a cell. Out, and potassium in. Because guess where sodium likes to sneak? Just like a sneaky kid wants to go where it isn't needed. It likes to sneak into the cell, and guess where potassium likes to go? Out. It's mostly because of concentration gradients. Okay? Are you with? Do you know what that is? Concentration gradient? Yeah? Okay. Where it's concentrated, it's going to try to diffuse to the area it's not concentrated from high to low. Right? Okay, so active transport systems, endocytosis, exocytosis, sodium-potassium pump. Okay, and we'll talk more about the sodium-potassium pump in a slide here, and then we'll take a quick break. Okay, so this just shows that it uses ATP, it turns into ADP plus a, free phosphate. Okay. Endocytosis, it's as simple as that. And the reason I put the picture in is because when it brings substances in, what it does with this plasma membrane is it will actually take part of that plasma membrane and it does what? It creates like a little vesicle for that substance with endocytosis. With exocytosis, we actually create, remember I talked to you about the vesicles that we create as we create like lipids and we envelop them and we send them out. We actually create a similar plasma membrane around this stuff that we will exocytose, and it'll melt into that plasma membrane and it'll spit that stuff out. So what are we doing if we're putting a similar plasma membrane around these structures?
Make an organelle.
You're making an organelle because you're doing what for the cell? Do you just want to let stuff in?
The membrane.
Yeah, you're making the membrane to protect the cell, right? Suppose this is waste that your cell needs to get rid of. We don't want it floating through the cell, you'll envelop it. Does that make sense? Okay. And there's an actual picture of it. I thought it was kind of cool. Okay. Endocytosis, a specific example of that is phagocytosis. Okay. Phagocytosis is the process by which we will ingest viruses, bacteria, or waste products in the body. So we have white blood cells which circulate throughout the body, which will turn into macrophages, which will phagocytose substances in the body. Do you think that you have a lot of waste products in your body? Absolutely. We're constantly building things up and breaking them down. So we need cells that will come in and digest those substances and infectious agents to get rid of them. Okay? So phagocytosis is the process, and macrophages are the white blood cells that do that. And it all happens through endocytosis. Resting cell. Oh, we have one person in. Oh, there she is, perfect. Resting cell, so I'm a resting cell, right? What concentration or what has the highest concentration of sodium? Outside or inside the cell? Outside, right? What do I have on the inside of the cell at a higher concentration? Potassium. We all get that, right? Okay. Do sodium and potassium sneak in and out? Absolutely. Are there other ions inside and out? Absolutely. We're just going to talk about these for now. Keep in mind that you have negatively charged ions. Sodium is an easy one to remember because the majority of the negative ions are chloride outside, and sodium chloride gives us what? Table salt. Okay, well, then we'll get into it, all right? Just remember, sodium out, potassium in, Na plus sodium, K plus potassium, all right? So the sodium-potassium pump senses when we have an imbalance of that sodium and potassium. So sneaky little sodium likes to sneak in, sneaky potassium likes to sneak out, but we also have situations where we'll have a major influx and outflux of potassium and sodium, and that's when nerves and muscles work. We're going to talk about in the future, okay? But our cell membrane can sense, I'm sorry I have my cell phone on, just so you know, my father-in-law is getting checked out, so if that rings and I gotta go, I gotta go. So, doesn't mean go, but all right, just give me a minute and I'll get back in my class. Okay, so sneaky sodium and potassium is gonna go where it's not supposed to be. Sodium in, potassium out. When it gets to a level that cell's charged, because don't forget, they're charged particles, gets out of the normal range, what will happen is you have the sodium-potassium pump. And the sodium-potassium pump is an active transport system. That's why we're talking about it lumped in with the active systems. It will regulate levels of sodium and potassium where they should be. So it'll pump sodium what? From inside the cell to out, and it'll pump potassium from outside the cell to in and reestablish that balance. Does that make sense? Okay. Sodium likes to sneak in because it likes to go from a high concentration to a low. Potassium likes to sneak out because it likes to go from a high concentration to a low. There's other reasons as well with charges, but that's the basic gist of why they do what they want to do. All right? Okay.
And you
No.
said you have like the potassium easily exit and sodium does not because of the size of the cell? Sodium does not.
Sodium, yeah, no, don't, just disregard that. Sodium does sneak in. Potassium likes to sneak out more. It actually is easier for it to get, but just disregard that. I don't want to confuse you. Sodium will sneak in. All right. As a side note, just for reference, okay, and I'm not necessarily talking about this last statement. Well, it is true as it goes inside and outside of cells, but this is true for us systemically. Just to put a little nugget into our head, where sodium goes, water goes. Okay, so it's true going into and out of cells. It's also true with our bodies. If we eat a lot of sodium, we're going to retain fluid. If somebody has high blood pressure, one of the things that they're told not to do is eat a lot of sodium, right? Because where sodium goes or salt goes, the sodium is a byproduct of salt, water goes. If you're retaining water, your blood pressure will go up. Are you with me? Okay. You can also tighten down your blood vessels, but that's a different concept. Okay. Same thing with cells. If they have a lot of salt go into them, guess what follows? Water will go in, right? So where salt goes, water goes. It's just a good thing to get into your head right now. Okay? Does that make sense? we're gonna jump ahead one slide. This may be hard for you to see in the notes, so you can move up front if you need to see this better. This is the sodium-potassium pump. This is the first step here. So we have that little purple people eater protein, right? And what happens is sneaky little sodium has snuck in, sneaky little potassium has snuck out, but the cell senses that it's become imbalanced and it needs to use a sodium-potassium pump to re-regulate those ions. 3 sodium engage with this protein pump and move out. Tell me what that charge is. 3 what? So sodium is positive.
So it makes cell to be more negative.
Don't jump the gun yet. You're going. You're stealing my thunder. Right? So 3 positive are going out, and as you so astutely recognize, only 2 positive are going in. There are negatively charged proteins inside. There's negatively charged chloride outside, right? So we have positives and negatives. So if we have positives and negatives inside and outside, and we're pumping more positive out than we're pumping positive in, 3 positive out, 2 positive in, we're getting a what on this charge or this cell membrane? What kind of charge on this cell membrane? Overall would be negative. Are you with me? Okay, do you need me to repeat it? Why? There's lots of reasons why that we're not going to get into. That's a whole semester of in itself. It's due to balances and charges and how this protein pump works. What you need to remember in a resting cell is it's negative. And one of the main reasons that drives that is the sodium-potassium pump, because it pumps 3 positive ions out, 2 in. Are you with me? And then you'll have negative charges on both sides. If you're not with me, raise your hand, because if you're not with me, I bet you somebody else isn't with me. Okay, all right. So now that you have that visual, if we go back to the notes, this should be easy, right? We have the movement of those charged particles, the 3 sodium out, the 2 potassium in, negatives also on both sides, which will give us a negative resting membrane of 70. Why? That's where it lands when it's resting. If you take 2 little electrodes, they actually used to use a squid nerve to test this because it was a nice big nerve with nice big cells, and they could actually put what's the word I'm looking for to test charges?
Amp.
Yeah, like an amp, right, to check the amperage of it. And they could actually see at rest it was negative 70 millivolts. Now different cells will be at different numbers. I will always go by this number, okay? So if you have a resting nerve or a resting muscle and you put 2 electrodes in there, that's what I was looking for, you can see that the charge is negative 70. And why? Because, okay, that's what they are at rest. And once again, sure, there's a lot more that's as far as we're gonna go with it. We're actually gonna go more, okay? Does that make sense? Any questions on that? We're gonna go over that concept again in the future. We go over it 2 or 3 times because as you can imagine, it's a very difficult concept, but it sets the foreground or the basis for all of neurology. Is anybody interested in neurology? Because this is where it begins. All right. All right, talking about fluids in general, okay? Isotonic, you've probably heard this. Isotonic solutions, I always remembered as being I am so tonic. Isotonic solutions match your body solutions. If you're going into clinical, there are several of them, okay? I don't know them, I don't teach them, right? But I can tell you that those solutions may match your own body unless you have a specific problem, like low glucose. They're gonna have to give you glucose or something, okay? But A solution that's isotonic will match your body's solution in a normal state. Okay? I should say in a normal state, because you can fidget with those solutions to fix people, right? Okay? Now, a hypertonic solution is going to have too much solute in it. If you've ever had a really salty meal, I forget, I think we had hot dogs the other day, and they were delicious. I think I had like 3 or 4, but there's a ton of salt. When I got up in the morning, my fingers were all swollen. I couldn't get my rings off, because where salt goes,
Water.
what?
Water goes?
And water goes, right? So a hypertonic solution will retain fluid, right? A hypotonic, and I'll show you a picture of what it does to cells, is going to be a solution with too little solute. For example, do you know what distilled water is? Okay, distilled water has no solute at all, nothing. So should you really be drinking distilled water? No. When you drink water, it has solute in it that matches your body's fluids better. Distilled water has nothing in it. I'll show you what it can do to your cells if you drink too much of it, okay? Osmolarity is the measure of total concentration of solute particles. I just don't get much into this because I feel like it's a whole separate lecture. Has anybody dealt with osmolarity in chemistry? No, we're not going to get into it. But just so you know, it's a concentration of those solute particles in that solution. You get milliosmoles in certain solutions and whatnot. You'll learn that when you get into your clinicals. This is a normal red blood cell. Okay. They look like, it's called a biconcave disc. Have you ever had a Werther's candy? They're those little caramels that have the indent in the center and they're thicker around the outside. That's almost exactly what a red blood cell is structured like. And if you've never had one, go get one, then you'll see what it's structured like. I should bring them into lab is what I should do. So it's a biconcave disc. In an isotonic solution, that's what a red blood cell will look like. What's happening in the hypertonic solution? So this is someone who has been given an IV with an excessive amount of sodium, which we wouldn't do that, you understand. But in theory, what's happening to that red blood cell?
Just pull the water.
It's what?
Pulling water out.
Pulling water out. Now, I always got confused with these concepts. So you know what I used to do on the exam? Even though your exams are on the computers, which I'm going to send you out this sign-up sheet, probably by tomorrow by noon, just so you guys know. I used to draw it. So I would put a picture of a cell, and with a hypertonic solution, I put a lot of dots around it. And that represented the salt, or whatever the solute was. And I knew that water would go from low to high concentration. Are you with me? So water would leave that cell, and that cell shrinks, right? This makes these cells not functional. They need to be a certain size. Guys, your red blood cells literally are almost the size of a capillary. And the reason it's so tight is because you can offload a lot of what? If the red blood cell's right near the small capillary, you can offload a lot of oxygen. If your red blood cells shrink, you can't offload it because it's not touching your capillary walls. Are you with me? Okay. On the other side, if we put it in a hypotonic solution, which means that you have little to no solute or salt, water will go into the cell. So if you drink distilled water, this is what happens to your red blood cells. And guess how big they'll grow? Not real big before they do what? Breaks. Okay? So we need to keep our solutions isotonic. And there's all sorts of regulations in our body to help us regulate that. All right? Or sense it. You see why it's important to have those isotonic solutions too. Right? Any questions on solutions? Right. Internal structures. I'm gonna kind of move through these quickly because I feel like these are point and shoot stuff. There's a silly song on YouTube that you can, if you pull up the PowerPoints, watch. I'm not going to because I want to make sure I cover all the basics today. So let's go through these little organelles and what they do. Here's a nice visual of them. Let's get right into it to make sure we get through everything that we need to get through today. Most of this, have people had cell bio or basic cell bio or general bio recently? Okay, so if you have not, and I'm not making sense, please let me know. I'd be more than happy to review stuff. Now the nucleus is going to be that center part of the cell, and I call it the brain. It's going to contain DNA. DNA is important because DNA is what will form the chromosomes when we replicate cells. It'll double itself, it'll form chromosomes, and then it'll split half of its DNA to each new cell, so we have a brand new daughter cell. We're going to talk a little bit about that at the end of this lecture. That DNA, though, is also very important, guys, because it gives us the template for what? Yeah, it gives us the template for proteins. And what do proteins make in the body?
Like the building blocks of
Yeah,
everything.
like everything, pretty much, right? It builds the structure, protein does, of the body. So in the DNA, in the nucleus, we have the template for every protein structure in the body. Pretty amazing. We also use proteins for enzymes, for example, okay? Are you with me? Okay, so DNA is important to replicate to create mitosis, which we'll talk about the steps in a little bit, but it also forms a template for all the proteins in the body. So let me ask you this question. We have this big long strand of DNA in our nucleus, right? If you were to take the DNA from one of my cells, you can run it from like here around the earth twice or something. I have no idea. Huge, right? So, why do I have different types of cells in my body? Why is it if in every one of my cells I have that same DNA order, that's my genetic code, why do I have different cells in my body?
Only certain DNA is expressed in certain
Yes, only certain parts of that DNA is expressed in certain cells. Very good. We start as a little ball of cells, and those cells start to differentiate and move towards being organs through a very complex series of steps. And they do so because specific parts of those DNAs are turned on. Does that make sense? Okay. All right. Okay, so DNA, we went through that, helps to form ribosomes. Those ribosomes are where we form protein. Okay, so DNA will talk to RNA. We're going to go through these steps, I believe, in the next set of notes. RNA goes to ribosomes. I'll show you where those ribosomes are in just a minute. The ribosomes are where we make protein. Okay. So, with genetics, unfortunately, we can have changes in those genes that can affect us. Changes or mutations. These are some examples that you should know. Down syndrome, and I don't get into the specifics. Well, okay, just know that Down syndrome is an example of one. We'll get into the specifics of it in the future. For example, Turner syndrome, a female only has one X chromosome when she should have what? 2, and that creates certain characteristics for her. Klinefelter, XXY, in a male actually creates a more effeminate male. They'll have smaller genitals, they'll actually have the creation of small breasts, tall and thin in stature. Cystic fibrosis is a lung and digestive disease where you have excess mucus that you can't expel from your lungs. You also can't get out of your digestive tract. It's a very sad disease. Marfan syndrome is a genetic disorder in which somebody grows very, very tall with very, very long, thin fingers. And I mean very tall, like over 7 feet. Marfan syndrome happens when someone is young before their growth plates close, as opposed to another condition called acromegaly where they actually grow thicker. I can't think, there was a wrestler that had acromegaly. But anyway, so Marfan syndrome, very, very tall. Muscular dystrophy is a genetic disorder where the muscles actually can turn into fat and connective tissue. So these are just a really cursory overview. If I were to ask you questions about them, it would be just on these basics straight from the notes, because there's a lot more to them than this, all right? But just to give you some examples. So ribosomes are these little areas that we'll find on the rough endoplasmic reticulum. Do I have a picture of it next in the notes? I do. Okay, I have a picture of it in the notes. And this is where we create proteins. And those proteins contribute towards the structures of the body. Okay. The ribosomes will be outside of the nucleus on the endoplasmic reticulum. I've got a picture I'll show you in a little bit. Okay. Those ribosomes are on the rough endoplasmic reticulum. And it's called rough endoplasmic reticulum. You know what, I should show you pictures. Let me go back to this picture really quick, okay? I like pictures. So we have the nucleus, okay? And then we have the endoplasmic reticulum. And it's this series of structures that kind of wrap around. And this is where we have the ribosomes, where the protein is made. That is called rough endoplasmic reticulum. We have smooth, and it's known as smooth because it doesn't have those ribosomes on it, just to give you a visual. We're also going to talk about the mitochondria as plural. Mitochondrion is singular. This is where what is created. Anybody? ATP. This is all in the notes. I'm just giving you a once over. Rough ER, ribosomes. Golgi apparatus is this structure here. And this is where we will actually encase substances with that like pseudoplasma membrane that we will use to exocytose stuff. Okay. Those are called vesicles. I think I talk about that in a little bit. I think that's all I talk about, but just to give you some visuals. So, nucleus we spoke about, genetic disorders we spoke about. We looked at those ribosomes where protein is created. I'm going to talk about those steps. Endoplasmic reticulum wraps around, it's a folded membrane. Rough ER synthesizes proteins. Smooth ER helps to sort of put the proteins together, but we also will package lipids in there as well, and hormones. Okay. It's kind of like the preparation dock, right, before it goes to the Golgi apparatus to send things out of the cell. Does that all make sense? Yes, no? Okay. There's a closer picture of it. Okay. I call the Golgi apparatus the UPS of the cell, just trying to give you things to remember, all this crazy stuff. This will package substances. It'll create vesicles with the membrane around it that's similar to that plasma membrane, so it can go up, meld with the plasma membrane, and leave the cell. Okay. Some of those vesicles will be used to destroy waste. We'll actually create vesicles that'll have enzymes in them. So if the cell ingests something that is dangerous or destructive, we can actually use that little vesicle from the Golgi apparatus to engulf it and destroy it. Okay? Okay. I know this is a lot of material. Okay, a little picture of the Golgi apparatus. Mitochondria provides energy for cell in the form of ATP. We know that. I always think of that as the hyperactive child for children. There's a little picture of it. It actually has a double membrane. There's one on the inside and one on the outside, and the steps to create ATP are pretty complicated. We'll get into some of those next semester. Fat and glycogen. Let me just see how much more I have in this. The rest of this is pretty easy, so let's zip through this. We'll take a quick break. We also store fat and glycogen in cells. Why would we do? First of all, what is glycogen?
Sugar, right? Energy.
It's how we store glucose, right? So we store glucose in nice long chains called glycogen. So we'll store glycogen in cells, so we store energy for the cells, right? We store fat in there as well as a second source of energy. Okay. Fat cells never go away. Did you know that? Once you establish fat cells, they don't go away unless you have liposuction. That's why it gets harder to lose weight over time. You can shrink them, but once you establish them, you don't get rid of them. Just a little side note. Okay. So cell support and movement. There's a cytoskeleton, which is the framework of the cell. There's centrioles, which are used for cell division. I don't touch much upon those really. I do talk about cilia and flagella. Cilia we spoke about, those hair-like structures that are on top of cells that move things. Flagella we find on sperm. It's the only location that we'll find it, and it's what creates the motility for sperm. And we'll talk about how those function in reproductive in next semester. So, metabolism. So cells use energy and matter. Don't forget, cells are little living things. They're constantly building up and breaking down. And that process of building up and breaking down is called metabolism. Anabolism is the ability to build things up. The way I always remembered this is if somebody's in or taking anabolic steroids, they become really big because they're building up their muscles. Catabolism is breaking down things, because don't forget, we need to break things down. If we run out of glucose, we'll have to go into glycogen to get glucose. That is breaking things down. When we break them apart or break them down, that's known as catabolism. Somebody told me that sounded like cannibalism to them, and it helped them remember. I don't know. All right? But it's going to be breaking down larger molecules into smaller ones. So if we look at the bottom, what is this process? ATP minus a phosphate turning into ADP. Would that be anabolism or catabolism?
So,
What do you think?
that's smaller. So catabolism?
Did you just say cannibalism?
Catabolism.
Catabolism. Yep, I agree, because you're breaking it apart. How about the second equation? ADP plus phosphate turns into ATP. That would be anabolism, okay? Or anabolic. Does that make sense? Good. Metabolic syndrome. Okay, 2 more slides and we'll take a quick break. So this is rampant with, unfortunately, increasing levels of obesity in our society. We're seeing metabolic syndrome more and more. And this is a confluence of at least 3 symptoms that combine together, and when they do, you tend to have a lot more health problems. So in waist of 40 inches for men, this means above a waist of 40 inches for men and 35 for women. Triglycerides of 150 or higher, those are your bad fats. HDL is actually your good fat. We're going to look at these in the future, but an easy way to remember is H is happy. So those are your good fats that actually have clear fats from your body. So that's 40 milligrams per deciliter or below. A blood pressure above 130/85. Those numbers always change, but we'll just go with that number. And a glucose of 110 or higher. So if you have 3 or more of that, you tend to have compounded symptoms or problems. Cardiovascular problems, you tend to have more issues being healthy, more cancers, etc. Okay, but we're seeing a lot more of this because of weight gain, unfortunately, in our society. I blame it on video games. All right, So just finishing up this last little slide, because glucose is super, super important. Glucose is going to be used to create the energy for our body. It's the primary source. You can use lipids, which are what? Fat. You can use protein, but the most efficient is glucose. When we have glucose and oxygen, we can make a ton of energy. So we need to ingest glucose. We also will store glucose. We store that glucose as glycogen. We store glycogen in two major areas of the body. Skeletal muscle. Skeletal muscles use tons of glucose because they are what is called highly metabolic. They use a lot of energy. The second place we will store is the liver. And we store glucose in the liver for that rainy day, okay? So, liver likes to share. If you don't eat enough glucose, we'll actually tap into those glycogen reserves in the liver. Skeletal muscle doesn't. It won't share. It uses it for itself, okay? So that's where we like to store glucose as glycogen. Now, if we ingest a ton of glucose, we'll use it for energy. We'll then store the excess glucose as glycogen. If we've eaten like a sundae and we have tons of sugar, the last thing that happens to glucose is it will be converted to fat. Okay, so energy, glycogen, stored as fat. All right, just so you know those steps. Okay, second form of energy, fats. Third form, which we really don't want to use, is protein. We don't want to use it, first of all, because it's very inefficient to use as energy, but protein creates what again? Structures of the body. If you start to use protein for energy, you will actually start to eat away at your muscles. We don't want that, okay? That's why it's important to have a nice balanced diet. Any questions on that? Okay. So, cells. One second. Do you remember what nucleotides are? I had it pulled up, but of course I... There we go. DNA versus RNA. Let's look at this. This is in chapter 2. Okay. You don't need to flip back to the notes if you don't want to. You can just watch. Okay. So, nucleotides. When we have DNA, that is in the shape of a what? Everybody knows the name of this. Double alpha helix, right? Which is like a really long ladder. And the sides of the ladder are sugars and phosphates. The rungs of the ladders are what? Nucleotides. They're also known as nitrogenous bases. Are you with me? So DNA has that double alpha helix with the nitrogenous bases or nucleotides as the steps. And they are adenine, thymine, cytosine, and guanine. Is this all ringing a bell now? Okay. DNA will talk to RNA to create proteins. Are you with me? So RNA is almost like a single strand. The only difference is RNA will have uracil instead of what? In DNA. Instead of what?
Thymine.
Thymine. Are you with me? So RNA will have a long strand, and it'll have, adenine, uracil, cytosine, guanine, the whole length of it. And the order of those nucleotides or those nitrogenous bases determine the protein it's making. It's forming the template. Are you with me? So a little strand of RNA comes in and gets the order of the nitrogenous bases, or the nucleotides are the same thing. I just know I use both of them. Gets the order from the DNA in the nucleus. And then it leaves the nucleus to go where? The ribosomes. So it takes the template for that protein and goes to the ribosomes, and the ribosomes will put the protein in order. Are you with me? So let's do it one more time. DNA has the template for proteins in its nitrogenous bases, or its nucleotides. It'll actually separate a little bit. RNA comes in, gets the template for that protein. RNA leaves the nucleus and goes to the ribosomes. The ribosomes will actually put the protein together. The protein will go to the Golgi apparatus. It gets encapsulated, it gets spit out. Are you with me? Anybody not with me? You can also email me if you have a question. Okay, but I wanted to go back to this screen just to refresh your memory on those bases. So if we go back to this, okay? Don't drive yourself nuts with this, all right? 3 of those bases are a triplet. A triplet represents an amino acid. This is as far as we're gonna take it. Could we take it more? I couldn't, but if you went into biology, sure, they could take off. Just know that 3 bases are a triplet. That triplet represents an amino acid. Okay, what I do want you to know is that amino acids, when you put them in a certain order, create a polypeptide, which is a protein. Okay, so what I want you to take from this is that amino acids are going to form proteins. And where do you get amino acids from? I'm looking like in the whole picture. I just had some. Get it from your food, right? You get it from your food. You're going to break down the proteins in your foodstuffs into amino acids. Amino acids are the building blocks for proteins, is what I'm getting down to. You're going to store those amino acids in your bloodstream, right? And then send them into cells when we got to make proteins. Not actually always in there, but you get the point. Are you with me? You sort of get it? Okay? Yes? Okay. All right. So, polypeptide, just know amino acids are going to form proteins. We're not going to get in, because polypeptide is like different from a protein, don't worry about it. Amino acids are going to form your proteins. Proteins are the building blocks of the body. Okay, here's a little picture. So you have transcription versus translation. Transcription is when DNA talks to the RNA and gives it its template for the amino acids. Okay? RNA then goes out to the ribosome and it translates. I always think of translation because it's like you're translating to a different language, right? At that point, I don't know if that helps you. You're going to form the polypeptide, which is going to be your protein. Okay, just to give you a visual. Yes, no? Yep, okay, good. How do cells replicate? We're going to look inside that nucleus and go through some very, very basics of how they replicate. Okay? So we have interphase and cell division. During interphase, your DNA is going to replicate itself because in order to undergo mitosis, you need to create 2 identical what? Cells. You'll replicate the DNA, so then you can do what? Cut the cell in half, and now you have 2 sister cells. Are you with me? Okay, so during interphase, you're going to replicate the DNA, and then we're going to talk about mitosis, which is the cell division. Okay. Mitosis is obviously very important because cells do what? Some cells get what? Why would we have mitosis in the body?
To grow.
Hmm? To grow, to regenerate, to create new tissues. Think about all the cells that you wear off your skin. You've got to create new cells. That happens through mitosis. There are certain cells, though, that don't mitose. Okay? Cardiac cells, nerve cells don't undergo mitosis. This is why if you have central nervous system damage, it's permanent. Okay? This is why if you have a heart attack, the surrounding cells pick up. Okay? Skeletal muscle doesn't mitose, just to give you some examples. Okay? But a lot of the cells in your body do. Oh, I do put them up there. Okay? Nervous, skeletal, cardiac. Okay? So you have chromatin, which is the long string of DNA. It's actually wrapped around proteins just to simply try to give it some shape, because it really looks, when the cell is not dividing, like a big bowl of spaghetti. And these histones are just simply proteins that help to wind it up a little bit. That's all we'll talk about with those. It will, when it's ready to replicate, after it's duplicated itself, form chromosomes. Okay? And then, because that'll help to kind of organize it so it can be split, and we could give half to one and half to the other. Okay? And it forms what are called chromatids, which I'll just show you in this next picture. The chromatids are just the arms of it. One half will go one way, one will go the other way. But what I like with this picture is just looking at the string of DNA. There's your double alpha helix. Here's those histones, or the proteins that it wraps around, right? And then it coils on a string, and this string, forms this little dot right there. It's just amazing how much information we have on these chromosomes in every cell that we have. Okay. Now the steps of mitosis. So we have interphase, which I mentioned earlier. This is resting from dividing, but this is where DNA replication will happen. And you'll unwind those chromosomes, the DNA will completely separate, and you'll replicate each strand. So if you replicate each strand, you're going to form two identical strands that you can divide to each sister or daughter cell, I should say. All right? So you'll match them. Did I go over that in chapter 2? So when we're talking about those nitrogenous bases, adenine, thymine, whatnot, right? So I'm just going to switch the letters because it'll be easier. A matches with what? T. And then G matches with C, right? Unless you're talking about RNA, and then you'll have uracil instead. Do you guys understand that? Okay. If you don't, come see me and we'll talk about it. So with that DNA strand, you'll replicate the matching nitrogenous base, or your nucleotide, right? So you have two matching strands. And then one will go to one cell and one will go to the other. Everybody good with that? Yeah? If you're not, come see me, okay? Then it'll rebond, form chromosomes, and then it'll get ready to replicate. I thought this was a cool picture showing how this is the original strand of DNA. If you're not catching that, you'll actually find bubbles that form along the full strand of the DNA because it's so long. So it doesn't like unzip from one end to the other. It forms bubbles, so they happen almost simultaneously, and it'll split, replicate, and then it forms two identical strands. I don't know, does that help you visually, or maybe, I don't know. Are you with me with what this is showing? Yeah, if not, come see me. But if we started unzipping from one end to the other, it would take forever. Instead, you have all these bubbles form the length of the DNA, so it can happen quickly. You can replicate it, you have two identical strands. Okay. So there's the chromosomes with the centromere, which is its center part, and the chromatids, which are the arms. I'm not going to talk too much about that. More what I want you to know, and this is kind of a cool little note you could read on your note. I won't test you on it, but about the numbers of what happens here. I just thought it was interesting. But I want to move on because I know I only have half an hour with the videotape. Karyotype as well. This is actually how they document those chromosomes. You could read through that on your own. I won't test you on it. Okay? Let's get into the cell division itself. So mitosis is the division of the nucleus. Cytokinesis is the division of the cytoplasm, which is the last step. The different phases. I could never remember this, so I tried to put some buzzwords in for you. So prophase, pro-chromo. This is when the chromosomes will form. Okay, your DNA is replicated. It's ready to form chromosomes. This is where it happens in prophase. Okay. Centrioles are in place for the mitotic spindle. The centrioles, I told you we're not going to talk much about it. They form the basis on which all those chromosomes can actually align between them. Chromosomes will form. It moves into metaphase. I always remember this as me on the equator. All of the chromosomes move onto the center of that mitotic spindle, and they're getting ready to be pulled apart. And they split, anaphase. So the chromosomes split, they will move to opposite sides of that mitotic spindle. Okay. And telophase is when the cell splits and it undergoes cytokinesis. So I don't know if those little buzzwords will help you or not, but I can never remember the steps. I thought they were pretty helpful. Okay. Then you have two daughter cells. Those were out, they do it again. Those were out, they do it again, right? Okay, constantly in cells that are regenerative, regenerate those cells as needed, all right? Does that make sense? Can you sort of picture those steps in your head? Good. That's when you know it, if you can actually see it in your head. These are some phrases that you should know and understand when we're dealing with cells, okay? So stem cells are big buzzword, right? And stem cells are undifferentiated cells that when you put them into a certain grouping of cells can take on the attributes of those cells. It's hot research, if you're into research at all. People are using them in joints to heal joints. People are using them in the nervous system to help the nervous system heal itself, even though you can't make more cells. It's a pretty hot topic right now. You can actually get stem cells from your own body. You can harvest them from different areas of your body. So, pretty interesting research. Cell differentiation is the ability, remember I told you in every one of your cells you have the same genetic material? Cell differentiation is when the genetic code for that particular cell is turned on. So as you develop as a fetus, you'll reach a point where the organ cells start to turn on. That is due to the cell differentiation. And it's when that specific part of the genes gets triggered. That's the magic of it all, right? It's pretty amazing. Apoptosis, does anybody know what this is? People love this one. Programmed cell death, right? And this happens when your body senses that that cell is going wrong, precancerous, abnormal, and it actually will break down and its components will be digested by your immune system, by those macrophages we spoke about, okay? Hyperplasia is an increase in cell replication. If you have a callus on your foot or your hand, those are cells that are hyperplastic or undergoing hypoplasia. Hypertrophy is an increase in cell size. You should put that down. Hypertrophy, just like the word hyper, God bless you, and the word trophy. Hypertrophy is increase in cell size. So if you go to the gym and you start lifting weights and you build muscle, you're not building new muscles, you're creating bigger muscle cells. Okay, hypertrophy. Atrophy is when you don't use your muscles. if you've ever had a cast and you take it off, and your arm or your leg is so much smaller than the other one, that's because those muscle cells have atrophied. Once you start using it again, they'll grow back to the proper size. Okay? So atrophy is when the cells shrink from disuse. All right? Does that all make sense? So just some buzzwords that you should start putting into your head to know. Any questions on that one? Hypertrophy is hyperentrophy. It's an increase in cell size. As opposed to hyperplasia, which is an increase in cell numbers. These are pictures of the different phases you can look through on your own. We're not going to sit and walk you through them. All right? Once again, sometimes I think pictures are helpful. If you can picture it in your head, you know it. All right?